Hi and welcome to the first of these videos which I'm recording as a supplement to my introductory biochemistry lectures. I'm going to look at a paper published in 2011 in which Hanczyc reported the production of some simple structures called protocells, contain a very simple metabolism. We're going to use that to link to some of the concepts we talked about in the earlier lectures as well. I say this is a supplement. It's worth mentioning here that my biochemistry lecture 10 will only appear in this format. So I won't be giving guinea pigs, humans and carrots to 100 people in the lecture theatre, but only in this YouTube format, which will be a new development. Okay, there's going to be 3 or 4 of these videos, I guess. There's a limit to how much I can upload to YouTube, about 15 minutes I think. So in this one we're going to talk about where the paper is. We're going to look at the overall structure of the paper and talk about some general advice about reading it, and a little bit about what it says. We'll have a look at some videos. In the next video we'll look at the context for this, which is abiogenesis. I'll attempt to understand the origins of life and the origins of biological chemistry specifically. And once we've set the scene there, we'll go on to look in some detail at the chemistry of what was happening. We'll use this to link back to, as I mentioned, some of the concepts we discussed in the earlier biochemistry lectures, and ultimately we'll wander back to the paper and wrap everything up with a detailed discussion of what it's saying. Okay, so finding the paper, a bit different. It's not in Scopus or Science Direct. There's lots of good stuff in those databases, obviously, but there are other resources which you shouldn't forget about. This is in the journal Philosophical Transactions of the Royal Society B, which essentially produces reports of conference proceedings. So the paper's a little bit different to a normal research paper, you might see. Other useful non-Scopus Science Direct resources are PLOS 1. PLOS stands for Public Library of Science. So there's PLOS 1 and there's also PLOS Biology. Lots of interesting stuff published there these days. So don't just look in the main databases. Look around, understand what's relevant to your particular program. You can find the paper in the link there. It is available publicly. You don't have to pay for it or anything. It was part of a whole journal whole journal issue that was given over to a discussion of the early chemistry of life. Lots of interesting stuff there. There's an interesting paper on the early development of chirality and achirality specifically, so why we have L-amino acids in life rather than D-amino acids. May do one of these journal club video series on that a bit later on. As well as the paper, there's a useful data supplement which contains some of the videos which we're going to see in a bit. In fact, it contains more videos than the ones I'm going to show you. Though the videos we're going to see are actually linked back to their YouTube designations because they're a bit easier to find. There's also an audio supplement which is essentially a recording of the conference proceedings. One thing you'll find when you're reading papers is that sometimes you have to read other papers to understand what's happening, and that's very much the case here. The Hanczyc paper we're talking about here has a lot of the background in this earlier paper published about 4 years ago. So a lot of the background on how the experiments were done and the basic chemistry is there. So often when you're reading a paper, you do need to read around it, either in other papers or in textbooks or other reputable sources as well. I found this as part of a YouTube video channel called the TED Talks. The TED Talks are a series of short video talks given by subject experts. Lots of good science ones in there. Hanczyc is well worth having a listen to. So when you've watched this video, link over to the TED Talks channel and have a listen to what Hanczyc says. Then that'll help you put the rest of what we're going to talk about in context. So there's lots of good science stuff there. There's a good recent one on spiders and spider silk specifically. I can see me using some of that material in a future first year lecture, particularly the information about how the different amino acid sequence of spider silks relates to their physical characteristics like stickiness. Okay, reading the paper. As I mentioned, it's a conference proceeding, so it's a little bit different to a sort of research paper, for example. So it doesn't have the structure of abstract, introduction, methods, results, discussion / critical analysis. Essentially, it's a summary of a number of papers and other work that Hanczyc published, given to an audience rather than presented as a journal paper per se. It's a nicely written paper. Hanczyc is a very good writer and his English is very clear. There's a couple of things I would suggest you need to think about when you read all papers in general. One is make sure you understand what the title's saying. So there's basically three concepts there: metabolism, which I would hope you will understand, motility, and prebiotic structures, which may be terms you haven't come across. A good idea to make sure you understand what these terms mean before you start reading the paper in detail. In terms of reading the paper in detail, I'm not going to do a line-by-line analysis of the whole thing. I'm going to do a sort of helicopter view of the stuff that's on this page here, which is some of the simpler protocells that Hanczyc and his collaborators have produced. And then, as I say, link them back to some of the concepts we discussed in earlier lectures. Later on in the paper, he talks about more complicated systems, including systems using oil droplets. As we'll see, the systems we're going to talk about, the protocells, we're going to discuss here, are very simple, containing only a small number of substances which are well understood. When using oil droplets, it's a lot more complicated, and they're able to produce a lot more interesting behaviour. So if you're interested, read the paper and check out the videos as well. Another thing you want to consider about reading the paper is the keywords. There's keywords in every paper, and I've highlighted in red the ones that are associated with this paper here. Essentially, they're used for indexing functions. So, for example, if you were interested in the concept of chemotaxis, you could do a search in Science Direct or wherever, and it would come up with a bunch of papers, including this one. I'll mention briefly that is chemotaxis, not chemotaxis. Okay, anyway, moving on. The other thing I've done there is I've got a PDF of the paper, and I've gone through highlighting things which I think might need a little bit of definition for when I produce this video series. So, for example, I've highlighted the term Marangoni instability there. Not a term I've come across before, though I have seen evidence of Marangoni instabilities in various systems. But I felt I did need to sort of define what that meant, and we'll come back to that later on. I've also gone through listing the chemicals we used. There weren't many. The behaviour which we're going to see, which is quite complicated, was done as produced with a very small number of chemicals. Okay, so the takeaway message there is make sure you understand the keywords. Perhaps do a quick read of the paper, go through highlighting the stuff you think you're going to need to understand, look at the definitions, get your head around them, and then come back and read the paper in more detail. Okay, so I've mentioned this term a couple of times, and what you should do when you come across terms that are new is define them. If you aren't defining the paper, and you often won't be, you need to find a good quality definition. In this case, I had a look in the eBury electronic books database, came up with this short monograph on the ethics of protocells. So what this essentially is about is what are the ethical implications of producing artificial life. An interesting topic, which I'm sure many of the biologists in the future will perhaps find themselves involved in. Probably the food students as well, because of GMOs and whatnot, which is a slightly different area, but ethically have similar implications. But the point about this is that there's a good definition of protocells there, which I've quoted on the slide, and I've also referenced it back to the definition. So, another thing that the book talked about, which I've just mentioned, was the generic ways in which you can produce them. The gentleman there is J. Craig Venter, who you may have heard of in the context of the Human Genome Project, in which he was one of the driving forces. His research group is looking at taking existing organisms, stripping out the genetic material essentially, and replacing it with artificial genomes to produce organisms with novel characteristics. Hanczyc and other groups take what's called a bottom-up approach. They start with simple chemicals, which they then allow to self-organise. It's arguable that perhaps models better what was happening in the early history of life on this planet. But it's worth mentioning that neither route has yet achieved success. That is to say, we haven't been able to produce an artificial organism, but I have little doubt that it will happen in the next few years. Okay, Hanczyc's Protocells. Now, as mentioned, he's taken the bottom-up approach, which uses simple chemicals, and very few of them. I'll talk in detail about these chemicals and how their chemistry works in the context of this in a later video, but for now we'll just say it's an aqueous environment. Water is a prerequisite for life, and the system we're talking about here is in water. Little red bead there is a droplet of nitrobenzene. Nitrobenzene is a hydrophobic oil, so you drop it in water, it self-organises into a little sphere, keeping as much of it as far away as possible from the water. It's red because it's had a dye added to it, the dye redo, which is an azo dye. Also in the little nitrobenzene droplet is some oleic anhydride. Again, I will explain what this is in a later video, but for now what we need to understand is that this single substance, oleic anhydride, when it comes in contact with water, which it does at the boundary between the nitrobenzene and the water, will hydrolyze into oleic acid. Oleic acid is a fatty acid, and this hydrolysis of oleic anhydride to oleic acid has two consequences. One is it changes the surface tension around the droplet, which is what causes the movement. And the other one is once we have oleic acid produced, it's a weak acid, so it will partially dissociate, producing hydrogen ions. So the white trail you see in the image there is a trail of lowered pH produced by these hydrogen ions. So we've got a simple system here which has some quite remarkable properties. Let's fire this video up. Right, so this is the video. Remember, the red droplet is nitrobenzene containing a dye and oleic anhydride. The oleic anhydride is spontaneously hydrolyzing to produce oleic acid, which is changing the surface tension, and it's also changing the local pH as evidenced by the white trail behind the droplet. Okay, that was a simple system. Hanczyc then went on to describe a slightly more complicated system with a slightly more interesting behaviour. It contained exactly the same, except, as we'll discover in the video later on, a solution containing a different pH was added, and the droplet moved towards the pH, the changed pH. So here we go again. There's our normal droplet whizzing around. Whiz, whiz, whiz. And now we're going to add a solution containing a higher pH. And the droplet immediately heads towards it and also moves chemotactically towards the highest pH available, which will be the centre of the spreading droplet. So that's another sort of behaviour. Okay, that was part one. Have a look at Hanczyc's video and have a sort of read of the paper, and we'll come back to part two where we'll start talking about why we're interested in this area, the abiogenesis problem. Okay, thanks for listening. I'll see you later.